Hello, our lecture today, lecture number 38, is on the biology of aggression and emotion. And we've already taken a look at some aspects of aggression, maternal aggression in females, intermale aggression. But understanding this from a more comprehensive viewpoint and its relationship to emotion, I think, is important. And behavioral neuroscientists have historically been interested in uncovering the neurochemistry and the underlying neurochemistry, as well as the pathways that are important, neural pathways that are important for aggression. So that's what we'll be looking at in this lecture. So to be clear, psychologists have spent a good deal of time tracing just what happens when we're in a situation where there's a high level of emotion that appears, and how we go about labeling that emotion, and how that in turn influences our perception, our cognition of various situations that we're in. So this linkage then between aggression and emotion and what we actually experience, and how we go about labeling those feelings that we have has again historically been important in the field of psychology. So we're going to start out by looking at a little bit of history, by comparing two theories that really dominated this part of the study of behavioral neuroscience for a long time. We're going to be taking a look at what's called the James-Lange theory and the Cannon-Barth theory of emotion. So if you take a look at this diagram that you see here, this is an individual now that is perceiving a stimulus. In this case, it's a snake. And according to the James-Lange theory, which is the red arrows that you see here, when you perceive that stimulus, what is going to happen is you're going to experience a somatic change, a visceral change. This is an emotional expression that's going to occur. And once you experience that, then that in turn is allowing for this emotional experience, which in this case is fear. So again, as a consequence of the bodily response that to the situation that you're in this case, viewing a snake, then you become afraid. The Cannon-Barth theory is a little bit different, and again, same stimulus, that of a snake. We perceive that stimulus, and the Cannon-Barth theory says that you experience emotion after seeing that snake, and it is one of fear. And then that in turn causes these somatic changes, these visceral kinds of changes that you feel. But again, it's only after you have become fearful. Then what follows are these biological changes. So the Cannon-Barth theory and the James-Lange theory are really saying very, very different things about the development of emotion. Two other psychologists who have made major contributions in this area are Schacter and Singer in what is called the two-factor theory. To take a look at this diagram that you see here, if there's a threatening stimulus, this is a grizzly bear that's approaching you, you experience arousal, your heart pounds, you tremble, you start running away, and then you give this a cognitive label. And that cognitive label is, that is one scary bear, I'm afraid of it. And then the emotion of fear ensues. This stimulus then, in this case the bear, is triggering this physiological arousal. Your brain is interpreting that, and that is leading to this cognition. Okay, this change that occurs in terms of how you go about labeling that experience. The two-factor theory is saying that the emotion that we feel, that we are experiencing when we're aroused, is really determined by the environment. And it's those environmental factors that are really influencing, determining the emotion that we believe that we are experiencing. If a couple of examples here might help you to understand this a little bit better. Let's assume that there's someone who's pointing a gun at you. You become very tense. You start sweating. Again, the gun is being pointed at you, and then you label this. Okay, I am afraid. Let's take a look at, again, your feeling of being tense and sweating. You're looking at this beautiful girl, Sue. She's looking at you, she's smiling, and then you label this. And again, the emotion that you experience is, well, I'm in love. Okay, so again, the same kind of feeling, initial feeling that you have, but depending upon what you actually feel in terms of your emotion, is really going to depend upon how you cognitively label that particular experience. So again, this is the Schacter-Singer theory, which I think is the most popular view right now of how we go about experiencing emotion. So, we noted earlier that scientists in this area have been interested in exploring what parts of the brain are involved in emotion. And certainly the limbic system, the old part of the brain that we see here, and the amygdala, and other supporting areas around the amygdala, like the septum, for example. These have been considered to be very important areas in the regulation of aggression, in addition also to the frontal cortical area of the brain. So for emotion, for aggression, certainly the amygdala has been a focal point, but also the septum, the thalamus, the hypothalamus, and the forebrain have always been considered to be very important. When we take a look at, again, a little bit more of the history in this area, certain case studies kind of pop out as being important in this history. If you take a look at this individual here, his name was Charles Whitman, and this very famous case from back in the 1960s, the early 1960s. Charles Whitman, there you can see him on the day of his marriage, became estranged from his wife a few years after they married. And she eventually moved out and moved in with her mother. And Charles Whitman was experiencing some, very severe psychiatric problems at that time. And he was seeing a psychiatrist on a regular basis. He was also having difficulty in terms of coping with the separation and then later the divorce from his wife. He was an excellent marksman. He would regularly go to a shooting range in Austin, Texas, and use his high-powered rifle there. And he kept a very detailed diary, and the entries in his diary are very revealing. One day, Charles Whitman awoke and wrote in his diary that he was going to kill his wife, and his former wife, and he didn't really know why. He proceeded to take his gun, his rifle, place it into a large bag, and he walked 2 blocks to where his wife was living with her mother. He proceeded to kill her in cold blood and also kill her mother. He then walked back to his house, sat and wrote in his diary that he had killed his wife. He didn't really know why. And then he proceeded to take his rifle again to the University of Texas campus and go to the tallest point on campus, which is this administration building. It's simply called the Tower. And over the span of about an hour or so, he shot and killed 14 people, wounded 31 others. This really was one of the first mass shootings in the history of the United States. And he eventually was captured by Texas state troopers, and it was upon autopsy, he was shot and killed. And upon autopsy, it was found that he had a very large infiltrating tumor in the amygdala temporal lobe area. We don't know whether or not that tumor was responsible for his emotional outbursts and his aggressive and violent behavior. He was experiencing a lot of psychological problems, psychiatric problems at that time, which probably also contributed. But it's very interesting that that this tumor did in fact impact the amygdala and temporal lobe, because we now know that this is an area that's very much involved in aggression and emotion. Back at that time, this is in the 60s and early 1970s, 2 researchers, one by the name of Kluver, another by the name of Bucy, were experimenting with infrahuman primates and exploring what would happen to dominant-subordinate relationships when when there was damage to the brain. And the way in which they produced this damage was to perform electrolytic lesions in which they would damage the amygdala. And what they found was that male monkeys became very docile, fell to the absolute bottom of the social hierarchy, rarely, if ever, would defend themselves in an aggressive encounter. Indeed, this was, some of the first evidence that was emerging at this time that the amygdala very well might be an important coordinating area for aggression in infrahuman primates. Another researcher by the name of John Flynn, Yale University, conducted some fascinating studies with cats, in which he was able to turn on different types of aggressive behavior in cats, depending upon where he stimulated them in their brain. So, for example, this cat that you see here received electrical stimulation to the lateral hypothalamus, and when that stimulation was produced, Flynn found this very intense predatory aggression. That's a rat that you see there in the cage of that cat that's being stimulated in the lateral hypothalamus. And once the cat received that electrical stimulation to the brain, it would engage in this quiet biting attack where they suddenly would jump on, they'd pounce on the rat and deliver a fatal blow to the head and to the neck. Flynn conducted another study where this time he stimulated the medial hypothalamus and produced a very different type of aggression, one that he labeled as irritable aggression. So the cat now is arching its back, it's hissing. Again, there's a rat in the cage. It bares its teeth and then it attacks and it bites the rat. So again, 2 different types of aggression depending upon the part of the brain that is being stimulated with electrical stimulation, the lateral hypothalamus and the medial hypothalamus. So indeed, this is a very interesting piece of work. when we explore some of the other research that was going on at this time, again, I'm trying to look at this historically. A Spanish researcher by the name of Jose Delgado started using this device that you see here, which is called a telestimulator. I mean, it's a, something that's about 2 to 3 times the size of a remote control for a TV. And what he did was, in a classic study that he did with bulls, he also worked with other animals, infrahuman primates, like what you see here. But the study that he's best known for is this one where in Mexico City, that he has this very aggressive bull that's used in bullfighting. And he implants electrodes in many different parts of the brain. And there's a series of buttons on this telestimulator that you see here, again, which looks like this. And the buttons on the telestimulator correspond to a different part of the brain. Each button corresponds to a different part of the brain. So it can stimulate selectively different parts of the brain and observe the effects upon this bull that is placed on the opposite side of the bull ring and starts charging towards Jose Delgado. And when Delgado presses the button that corresponds to the amygdala, the animal's violent, aggressive behavior stops, and the animal becomes docile. And this indeed is a very famous experiment in this whole area, and it's one that, of course, suggests that this is an area that might be an important coordinating point for aggressive behavior. There's also a relationship between serotonin, the neurotransmitter serotonin, and animal aggressive behavior. It was noted back in the 70s, 80s, and parts of the early 90s that if you take animals and you isolate them for an extended period of time, like 2-3 week period of time, that it does a number of things. It dramatically elevates their aggressive behavior. If they're suddenly confronted with another animal from the same species, a male, for example, of the same species, and if we isolate a male for 3 weeks or so, what we find is that they become incredibly aggressive towards an animal that enters their territory. And what researchers also found was that the turnover, that is the activity, the amount of serotonin that's being produced in the brain, dramatically declines as a consequence of that isolation. You also find in what we call free-ranging studies, free-ranging animals that are in the wild, where they've taken a look at, let's say, rhesus monkeys or baboons, that the most dominant animals, the most high-ranking animals, have the lowest levels of serotonin in their brain. So again, we have this interesting relationship which suggests that there might indeed be something about serotonin that is important for aggression. And again, this figure here simply shows the major serotonin pathways in the brain, and these nuclei here, called the raphe nuclei, are a part of this major pathway that we see. And again, take a look at all the different areas of the brain that these serotonin pathways go to. And, importantly, to parts of the amygdala, as well as, parts of the cortex of the brain. So, this is very interesting work. This has been extended in part to work that has been done with human beings. If you take a look at what happens with individuals that have a history of committing violent crimes, one of the things that we know is that their turnover rates of serotonin are quite low. Some very good work has been done taking a look at this association, this correlation. Researchers have also found that young people who have a history of exhibiting aggressive behavior, they too exhibit low serotonin levels. There's important genetic contribution to this. In individuals that have this less active form of tryptophan hydroxylase, which is an enzyme, that's an enzyme that converts tryptophan into serotonin, that seems to lead to aggression. So again, they don't have as much serotonin, again, because of this enzyme that is not very active, not very efficient in terms of converting a tryptophan hydroxylase, converting tryptophan into serotonin. So they have lower than normal levels of serotonin, and they have heightened levels of aggressive behavior. So again, there's some, reason to think that what we see in lower animals may be related to what we see in the case of human beings. Email Carcaro has been doing very interesting work looking at the relationship between serotonin and aggression in individuals, clients that are individuals that have a short fuse. They're constantly getting into trouble. People, for example, that are convicted of things like road rage, where they simply cannot control themselves. Maybe when they're just in a slight car accident, or someone who's ahead of them on a highway isn't going fast enough, or someone passes them and they get angry because they pass them. These are all cases of individuals who cannot really control their outbursts. They can't control their emotions, and again, they're constantly getting into trouble because of it. Carcaro has been having success using the drug Prozac, which is clinically marketed as an antidepressant, but he's had success in lowering aggression in these people that have a very short fuse. And indeed, that's very interesting, because one of the things that we know is that Prozac increases levels of serotonin in the brain. So that seems to be working to lower these impulsive behaviors, these angry outbursts that we see. So again, it's very interesting relationships that argue strongly that serotonin is involved in emotion and aggression. One last interesting avenue of study. What you see here is a psychologist by the name of Jaak Panksepp, who for many years was interested in exploring positive emotion. Instead of negative, destructive kinds of behaviors, he was interested in what he labeled as being positive emotion. And this is something that he could solicit in a rat by simply putting a rat on its back and tickling its abdominal area. Well, when we begin to take a look at this, one thing that happens when you do this with a rat is they start to emit these vocalizations, this chirp-like kind of sound. And when you see this naturally in rats, for example, when they're playing with one another, exhibiting rough and tumble play, you get these vocalizations that occur that are very similar to what you see when you stimulate the abdominal area of a rat, these positive experiences. So what Panksepp did in this very interesting piece of work was he did this for 5 days, and he stimulated the abdominal area of the animal, and the animal exhibited a lot of the chirp-like kinds of noises that suggested that it really liked being tickled in that area. And what Panksepp found was that there was a production of a lot of new neurons, something that we call neurogenesis, in the hippocampus. And he also noted that there were increases in opioid receptors within the brain. So, here we have I think, an animal model for studying positive emotion instead of the negative kinds of emotion that ordinarily psychologists have been interested in exploring. So again, I think that these are interesting findings, and these animal models that are being used now in order to assess positive emotion have really helped us a great deal, I think, in taking a look at positive emotions as opposed to these negative kinds of aspects of aggression and emotion. And it's Panksepp's work that has really helped us a great deal in this area. So that brings us to a close in this area in terms of aggression and emotion. And in our next lecture, we're going to be moving on to some other interesting areas, namely the area of addiction, for example, and taking a look at drugs in particular, the whole issue also of laterality. Those are things that we're going to be exploring in some of our future lectures.